Hello everyone, in this video we'll be talking about gram staining procedure. Let's proceed. History behind the gram staining procedure. Gram staining is an experimental technique widely used in microbiological and other diagnostic laboratory to distinguish between two major groups of bacteria. Before the discovery of gram staining, the bacterial cell wall composition was not clear. In 1884, Hans Christian Gram suddenly discovered gram staining while he was searching for a technique or method to visualize the pathogenic bacteria from a tissue section of a patient whose lungs was affected and died of pneumonia. In his procedure, the first step he made a smear on a clear glass slide with the fluid sample and then heat fixed. And then he poured gentian violet or crystal violet solution over the smear on the slide. After that, his slide was rinsing with water, added potassium triiodide solution or Lugol solution or mordant to fix the smear with dye. Then he washed the slide with phenol to remove excess dye. In his experiment, he observed that certain bacteria retain the color of crystal violet or gram positive, and he found pneumococcus, while some species decolorized by the ethanol or gram negative. His intention of the work with this staining procedure was to perform on the pathogenic bacteria Streptococcus pneumoniae and Klebsiella pneumoniae. Gram remarked in his initial publication that I have therefore published the method, although I am aware that as yet it's very defective and imperfect, but it's hoped that also in the hand of other investigators it will turn out to be useful. In his procedure, Gram did not use in any counterstain, so it was the limitation of Gram's experiment. Few years later, German pathologist Karl Weigert added a final step of gram staining with safranin to visualize the gram-negative bacteria. The basic principle of gram staining. Bacteria can be distinguished between two major groups on the basis of gram staining because the group of bacteria having thick layer of peptidoglycan can retain the color of crystal violet stain and iodine with the dye form an insoluble complex and show purple color are called gram positive bacteria. While some other bacteria having the thin layer of peptidoglycan retain the color of safranin and shows red or pink in color are called gram negative bacteria. Procedure of gram staining. At first, take a clean, grease-free glass slide and make a smear on it and heat fix. Then add crystal violet primary stain and keep it for 1 minute and gently wash with distilled water for 2 seconds. Then add gram iodine for 1 minute and then again wash with distilled water and then add 95% ethanol for 30 seconds. Then again wash with distilled water for 2 seconds and then add counter stain safranin for 30 seconds and then again wash with distilled water and then observe under microscope by oil immersion. Two types of cells are seen here in the gram staining procedure. In the microscopic observation will be like one bacteria will show purple color that will retain the crystal violet color is gram positive, and another bacteria will show pink color means they will retain the color of safranin and they decolorized the color of crystal violet, so they are gram negative. This is how we distinguish between gram positive and gram negative bacteria by this experiment.